not lose the most important messages. We have called Professor Merlinder to tell us what to bring home. Professor Merlinder, you are the head of the Institute. Please enlighten us and make sure that we take what is worth going home. Roger.
Jens, thank you for that nice introduction. And firstly said that this was for me a very busy meeting, a very busy venue, for I did not know before what the talks would deal with, and I have to summarize now the Congress, and I will try to give my very best. So it's really not so easy to finally conclude such an informative conference day with 11 talks and 3 discussion sections. First, Rob van Tetering, thank you very much to you and all your colleagues for excellently organizing this venue today. Thank you for the invitation to Worms, the city of Worms, and in the name of all participants. Thank you very much. Give them a hand. Now I will try to sum up the day from a more scientific and technical point of view. The starting point was the talk of Olivier Armand, and he states that nematodes are the most diverse phylum of all animals in the world. For me it was new. In sugar beet there are by far more species than the normally discussed Heterodoxarchaea, such as Beta, Megalogini, Chypodii, Detilenchus, and others. And he stated, wherever sugar beet are in the field, you will find nematodes. Nicolas Minerva specified that infestation level enormously varies. For instance, in the Netherlands, he reported up to more than 1,000 eggs and larvae in reality, maybe sometimes more, or opposite, in Italy, less than 100 eggs and larvae per 100 grams of soil are common, beside the fact that the damage is high in Italy as well. And this damage can reach in sometimes more than 50% in sugar heat. Consequently, tolerant varieties are intensively used European and US-wide. Jim Stewart reported that Heterodoxarchaea intensively exists in the US as well, but surprisingly, the 250,000 hectare acreage in the Red River Valley failed, with expected 0 eggs and larvae. For me, it's an unclear fact. Normally, Mohammed is knowing all these diseases and pests. Here, 0. The reasons for it might be from a technical point of view, crop rotation, and the cultivation history, which might be a little bit different in the US than in Europe. In the discussion of that section, there have been, from my point of view, some really important messages. And they were first, that the genetic variability of Heterodoxarchaea seems to be high, and most important as well, stable. This might have highest evidence for low risk of resistance breakage. Later on, we have gotten a little bit different message. Second, the continuous cultivation of tolerant and resistant varieties led to much lower infestation than 10 years ago. Both facts demonstrate the breeding and agronomic relevance for resistance management and integrated pest management, which has to be realized European-wide in future. Within the next section, the first talk was of Jan Sells from this Vandaram itself. He clarified the wording. Resistance is the capacity to propagate or not the nematode population. Resistant varieties do so not. Tolerance is the capacity to tolerate the nematode attack, so tolerant varieties have a lower yield loss under infestation, but not a higher yield, as Klaus Bürgi later explained. The today's tolerant varieties are partially resistant as they propagate nematodes less than susceptible, respective more than resistant ones. In the discussion, there was a focus mainly on that what Jan Sells says, so I will give the information here. He clearly states that rhizomania resistance is still the number one in breeding, you asked him. And that the definition of non-infested fields is not trivial. Often less than 30 eggs and larvae were analyzed. However, the damage of susceptible variety was high. It is not to understand from a technical point of view. So this is of particular interest and importance for both breeders and farmers. Hopefully, different genetic resources can be combined in order to get more stable, partial, resistant, intolerant varieties. The next talk has been from Christian Jung, and he mentioned that the genetic source of complete resistance comes from Beta patellifolia procumbentes, while the tolerance gene comes from Beta maritima. The genetic basis of both, he states, is very complex. It seems to be more than it seems to be a gene region more than a specific single gene, so very complex. The introduction of resistant genes normally failed as the different species cannot be crossed. But the HS-PRO1 gene was the first plant gene being completely sequenced, a pioneer performance of breeding research worldwide. Elmar Reimarkers states that there seems to be a cross-tolerance of Heteroda shachti tolerant varieties to Heteroda viti as well. Interestingly, other interactions exist as well. A high infestation with Heteroda shachti increases first the damage of Verticillium dahliae, so tolerant varieties prevent symptoms, and second, magnesium deficiency can be higher, the higher the infestation of the nematodes is. After the dinner, Mark Stevens gave an overview of thresholds across Europe, and that was really interesting. They are very diverse, not only in relation to eggs and larvae, however also in relation to more unspecific parameters and diverse parameters. Surprisingly, the UK colleagues use a different definition of tolerance and resistance. This all, ladies and gentlemen, demonstrates, positively expressed from a personal point of view, the enormous diversity of nematode research. The standardization is not easy, however, needed more than ever and tackled in the final discussion again. Marc-Richard Mollard and Sylvain Flournet reported from research on resistance management extensive trials in France. A resistance breakage was detected in the greenhouse for what they called major resistant varieties, in other words, complete resistant varieties, but generally this was not found in the field. However, first incidents for breakage occurs in Italy and Turkey. The message is that sustainable resistance management should be focused on a consequent, consecutive cultivation of tolerant and resistant varieties. Matthias Daub contributed that crop rotational aspects have highest importance for nematode management. The cultivation of oilseed rape is less critical than seen before for the cultivation, but there is a clear increase of the nematode population by volunteer oilseed rape directly after harvesting the oilseed crop. The propagation intensity has a clear relation to the initial population density. The relative effect is more or less the same for all variety types, the tolerant ones, the susceptible ones, and the resistant ones. Klaus Bürgi presented activities in extension service in southern Germany with relation to nematode control and in general. For an efficient work, one need, besides a clear reliance between grower and industry, a definite analysis of the problem, field trials, field demonstrations, and a clear research approach. Here, near Worms, this is a history since 60 years, and maybe another reason to have such a venue being held here. The population density in the subsoil has to be taken into account as well, Klaus Bürgi focused on. However, when nematodes are analyzed, tolerant varieties should be cultivated. In other words, threshold 1 egg or larvae. These varieties, the tolerant ones, showed enormous breeding progress in yield and performance with and without infestation the last 10 years and may play a major role in the enormous yield increase in general we see across Europe the last years. In the final talk, Uwe Luschkel and Cliff Watrin gave an insight to something new, the biological and chemical control of nematodes. The results are not so clear so far. However, it would be of highest interest to get further control measures for nematodes in future, even if a breakthrough is not to be seen in the short run. The discussion of the last section focused a lot on the last talk, and I want to highlight it in relation to all the others, the other statements, which focus a little bit more, let me say, for all the talks from lunch on. First, the clear need for standardization of terminology and analysis in nematode research. And I would say that is really something which we have to bring forward altogether. Second, the biological control seems to have a very broad efficacy against diverse species. And third, tolerant varieties should be cultivated independently from the initial population density, as you said. To sum up, today we got the most powerful information on nematodes which should-be researchers could offer in the world. I think so. It was an excellent opportunity to combine facts from agronomy, from breeding, and from science. I want to highlight the insights of the complex interactions and their consequences for breeders and for farmers. At the very end, thank you to all speakers, session leaders, contributors in the discussions, and to you, the participants. Furthermore, the interpreters are fit. Give them a hand, please.